Inside the human body lies a physical network of blood vessels so incredibly extensive, it stretches for 97,000 kilometers. With such a massive, continuously branching maze, the body faces an immediate logistical problem, keeping the oxygen-rich blood perfectly separated from the oxygen-poor blood. In many organisms, like insects and mollusks, this isn't a concern. They operate on an open circulatory system, where fluid freely washes over internal organs, mixing together in large internal cavities. But humans and higher vertebrates rely on a strictly closed loop. Our blood is confined entirely within a continuous series of specialized biological pipes. Given the sheer physical size and high metabolic energy demands of the human body, this strict compartmentalization is required to deliver oxygen fast enough to keep our tissues alive. For centuries, the mechanics of this system were misunderstood. Many early physicians believed blood was constantly being manufactured in the liver and then entirely consumed by the body's tissues. That understanding shifted in the early 17th century when an English physician named William Harvey began studying the beating hearts of living snakes. Measured observational. Looking at this original 1628 publication, Anatomica de Motu Cordis et Sanguinis, we can trace the exact moment Harvey proved that the heart acts as a pump, driving blood in a continuous circular motion, rather than a slow ebb and flow. By establishing this circular flow, Harvey provided the evidence to analyze the body as a pressurized mechanical system. Harvey's proof established a physical framework for blood flow, allowing the body's vessels to be analyzed as a calculated mechanical circuit. The physical journey through this loop starts the moment blood is pushed out of the heart and enters the arteries, known in Marathi anatomy as the mani. Because they take the direct, immediate force of the heart's contraction, arterial walls are built thick and highly elastic. They have to rapidly expand and recoil to handle that extreme outbound pressure. As a general rule, these thick vessels are dedicated to carrying pure, oxygen-rich blood away from the heart and out toward the extremities. There is exactly one biological exception to this layout. The pulmonary artery is the sole outbound pathway that carries oxygen-poor blood, routing it directly into the lungs for a refill. Together, the arteries function as the body's high-speed, high-pressure outbound highways, built specifically for rapid delivery. Those high-speed highways eventually narrow and branch out into their terminal endpoints, the ultra-thin capillaries, or keshvahenya. Here, the robust vessel walls drop down to a microscopic thickness, creating a permeable boundary between the bloodstream and the surrounding tissue. This structural thinness allows for a simultaneous two-way transaction. Oxygen and nutrients filter outward to feed the cells, while cellular waste and carbon dioxide are pulled inward into the blood. This quiet microscopic exchange is the functional reason the massive 97,000-kilometer network exists. Once that transaction is complete, the now oxygen-poor, waste-filled blood transitions out of the capillaries and into the veins, or shira, to begin the return trip to the heart. The venous system faces a severe physical challenge. It has to push heavy fluid upward from the legs and arms against gravity, and it has to do it without the initial high-pressure push of the outbound arteries. The mechanical solution to this problem was identified by an Italian anatomist named Hieronymus Fabricius, William Harvey's teacher. He observed a series of one-way mechanical valves built directly inside the lining of the veins. As surrounding muscles compress the veins, fluid is forced upward, pushing the valve flaps open. The moment gravity attempts to pull that blood backward, the downward pressure snaps the flaps securely shut. Without these simple structural mechanics locking the fluid in place step by step, the closed loop would simply collapse under the weight of its own contents. The pipes provide the structure, but the system relies on the specific properties of the fluid traveling inside them. Looking at this textbook's Raktadrov section, 55% of blood volume is a pale yellow alkaline liquid, blood plasma. Chemically, it's 90% water, a solvent carrying dissolved salts, glucose, and hormones. A crucial 7% is functional proteins, particularly one called albumin. Albumin performs a technical job. It regulates the osmotic pressure inside the vessels. By maintaining the right concentration of solutes, it ensures water stays inside the pipes and doesn't leak out into the surrounding tissues. The blood fluid is a chemical delivery system, calibrated by its composition to operate within the pressurized constraints of the vascular network. When we pull back from the microscopic proteins and individual valves, the true scale of the network comes into focus. All 97,000 kilometers operate together as one unified machine. The body keeps pure and impure blood separated through continuous dynamic motion. It relies on the sequential integration of high-pressure outbound arteries, microscopic exchange capillaries, and valved return veins. This precise mechanical routing and chemical sorting happens relentlessly, moving volume every single minute of our lives without a pause. Our survival depends on this consistent pressure within the highway, a structure mapped and verified by researchers over 4 centuries ago.